trained as a medicine in hepatology, became consultant hepatologist, finally professor of liver medicine of the University of Newcastle. He's very well known for his work in liver disease, particularly in fatty liver, steatosis, steatohepatitis, in the definition of the term steatohepatitis and how this disease progresses, and more recently about gene associated to the development of the disease. He's been editor, he's still co-editor of Journal of Hepatology, which is the, together with Hepatology, published in US at the top 2 journals in hepatology in the world. One of the things I really envy him is that last year in the Olympic Games, he was selected to carry the torch in Newcastle. The only scientist. I think it's fantastic. So he was carrying the torch in Newcastle. He said that there were 3 people selected, 2 were students. He said that he was the only senior faculty that could run. But
That's how I think
there were more merits than that. Thank you, Chris, for being here.
All right, thank you. So it's a great pleasure, Jose, to come to Bilbao and give a talk. I've not been here before, and I'm already very impressed with the buildings here and the view of the city I got from the aeroplane as we came into land. So I have 2 main interests in hepatology, both very related, really. The questions of why most obese people or people with insulin resistance get fatty liver and nothing else, and why most heavy drinkers, which will be the talk this evening, get fatty liver and nothing else, no matter how much they drink or no matter how overweight they are or how insulin resistant they are. And yet around 10% of each of those groups of patients get a horrible, nasty liver disease that ends up with cirrhosis and liver cancer. And what is it about those individuals that's different from the vast majority of overweight or heavy drinkers that just develop steatosis and die of cancer, strokes, heart disease, something else not related to their liver? And so really the subjects of both my talks are really trying to address those questions of identifying those patients, understanding the mechanisms why it is that they go on to disease and others don't, so we can start to design prevention and treatment strategies. So I'll talk a little bit about the risk factors for progressive non-alcoholic fatty liver disease, and by that I mean fibrosis, cirrhosis, liver cancer, as opposed to just steatosis. So if you take a bunch of patients with fatty liver disease across the range from simple fat through to inflammation, steatohepatitis, cirrhosis and liver cancer, and look to see what it is that's different about those with the more advanced disease, then you will get this sort of figures. So it's associated with obesity, it's associated with insulin resistance, it's associated with the presence of type 2 diabetes. The older you are, the more advanced disease you seem to have. And if you have the more features of the metabolic syndrome you have, which as you know is hypertension, dyslipidemia, obesity, the more likely you are to have advanced disease. So, and the key thing about these studies is that the more of these features you have, the more likely you are to have advanced disease, NASH and fibrosis, rather than just steatosis. So we've known that for some time. It comes out in every single study. There's been very few follow-up studies done to see which fatty liver disease patients, if you take them at the start and then follow them up, which ones go on to get more advanced disease. And this was a meta-analysis of a number of follow-up biopsy studies that started with patients and then followed them up for around 15 years. And what it showed was that if you had inflammation here, then you were much more likely to develop with time fibrosis. So the number that didn't have advanced fibrosis fell gradually, whereas if you just had steatosis, it was very unlikely. So it seemed to be the inflammation, those with inflammation on the biopsy, that went on to advanced disease. So the question we need to ask is, why and who is it that gets the inflammation, which then leads to the fibrosis and the scarring. So this just illustrates, it's a very old study, but it illustrates a point very nicely. This is the data from Ian Wanless's study, which is nearly 25 years old now, where they simply did post-mortems on everybody who was dying, who had died in a particular hospital in Toronto in Canada. And what they were able to do, they were looking on that post-mortem liver as to who had fatty liver and who had NASH steatohepatitis. And what they found was that your chances of having NASH at post-mortem on a liver biopsy went up the more obese you were. So this is no obesity, some obesity and a lot of obesity, and they went up even higher if you were also diabetic. So that just confirmed what I showed you on the previous slide, obesity and diabetes. But look at this here. So even the most obese and the type 2 diabetics, only 18% had steatohepatitis. The rest had a normal liver or fatty liver. So it just illustrates this fact that these may be the risk factors, but most people with those risk factors are fine. They just have steatosis. So that's the question that we're trying to address. So I guess most of my talk and the discussion afterwards will be focused on genetics, because that's been my area of interest. But there have been some studies that have tried to say, well, perhaps it's something else about the patient. And obviously the beauty of environmental factors is that you can alter them. So if it's something about your diet, for example, you might be able to alter that diet and prevent the disease or treat the disease. So environmental factors that might predispose to advanced disease are very attractive because they're much more treatable, obviously, than if it's just your genetic background. It's very hard to study environmental factors, though, in most cases, in particular diet, as some of you may know if you've tried to do detailed dietary studies in patients. But there has been some data emerging over the years. And so in terms of trying to look at dietary factors, some of these, as you see, there's not that many studies. A lot of this data comes from one study by Musso and colleagues in northern Italy. But there's been a suggestion that if you take those obese patients with NASH on a liver biopsy and compare them with obese patients with steatosis, that those with NASH eat a diet that's lower in antioxidants, which fits with the idea that NASH might be an oxidative stress disease. Higher in total and saturated fat, which I guess is, again, intuitive, given that this is saturated fat we know is the substrate for lipid peroxidation, which is thought to be involved. A diet lower in carbohydrates and a diet lower in the protective polyunsaturated fatty acids and higher in the more dangerous polyunsaturated fatty acids, which lead to pro-inflammatory mediators such as eicosanoids. And then there's been a lot of evidence recently, particularly coming out of the North American literature, that it may be those patients who eat a diet high in fructose. And we might be able to talk about reasons for that later on in the discussion. So not that much data, but there is some evidence that your diet might influence your risk of getting NASH and advanced disease. There's been a lot of work in the alcohol field recently about caffeine being protective against fibrosis. And again, there are a number of potential mechanisms. For example, caffeine is metabolized into theophylline, which is a phosphodiesterase inhibitor, a well-known anti-inflammatory. And there's now been the first study done in NASH by Stephen Harrison's group, showing that the more cups of coffee, caffeinated coffee you drink, the less likely you are to get fibrosis. And that makes sense because it's been shown repeatedly in alcohol studies. Interestingly, in terms of this is non-alcoholic fatty liver disease, and in most of the studies, including our own, you don't get into the study if you drink heavily, obviously, because then you've got alcoholic liver disease. But in most of these studies, they allow you to drink some. So in our studies, it's typically 3 units or 30 grams a day for a man, and 2 units, 2 drinks, or 20 grams for a woman. And when, in a number of different studies, they've looked to see whether those NASH patients who drink something, as opposed to those who drink nothing, it would appear consistently that those who drink some get less advanced NASH than those who don't. And that might seem slightly against what you would think, as you might think that alcohol and obesity would be synergistic and give you more disease. And there is no doubt, as I'll show in the data tonight, that if you are overweight and drink heavily, that those two things are additive. But if you drink a little bit, it would appear to be protective. And that, I was quite surprised by this until I looked at the other literature, and it's been known for a long time, for example, that small amounts of alcohol protect you against diabetes. They protect you against insulin resistance. And it seems that low amounts of alcohol have an anti-inflammatory effect, lowering cytokines such as TNF-alpha. And it may well be this anti-insulin resistant effect of small amounts of alcohol that's protecting these patients against fibrosis, if you believe, as seems to be the case, that high levels of insulin and insulin resistance are one of the factors driving fibrosis. Another idea has been perhaps it's those obese patients that have obstructive sleep apnea. So these patients that are so overweight, they get fat in the back of their throat and their mouths collapse, nasal passages collapse during sleep, and they have episodes of profound hypoxia, low blood oxygen, during sleep. And the idea was perhaps it's those hypoxic episodes that trigger the inflammation. And there's now been several studies that have shown those obese patients that have obstructive sleep apnea, defined by formal sleep studies, measuring how often during the night they stop breathing and their blood oxygen levels drop. They're the ones who get NASH. And again, the attraction of that is that it's treatable. You can treat obstructive sleep apnea with something called CPAP, continuous positive airways pressure, which keeps the airways open during the night and stops these oxygen drops. And we're waiting for the first studies to see whether, for example, giving CPAP to OSA patients with NASH starts to improve their liver blood tests or even liver biopsy. And then finally, as I'm sure you're aware in an institute like this, there has been increasing interest in whether it's something about the microbiome that we have, which we now understand is fairly individual to us. Perhaps it's the gut microbiome of a particular obese patient that not only predisposes them, we now understand, to obesity, but it might predispose them to NASH. And I'll show you some data on that in a moment. So they're the sort of ideas. Might be diet, might be the amount of coffee you drink, might be small amounts of alcohol that's protective and the abstinent patients are more at risk. It might be the sleep apnea patients, and perhaps it's those with a particular subtype of gut microbiome. So I'll just spend a minute on the microbiome and NAFLD. Why do we think this might be important? So in the 50s and 60s, if you were obese and wanted obesity surgery, you got something called a jejunal-ileal bypass. And that basically created a blind loop in your bowel which bypassed your jejunum and didn't allow you to absorb food. That blind loop became a fantastic site for overgrowth of gut bacteria. All of these patients, all of them, got NASH and fibrosis and cirrhosis, and the operation was abandoned eventually because it was seen to be too dangerous for the liver. So there was the first evidence, if you like, that if you put high levels of gut bacteria into an obese patient through creating this blind loop, you will create NASH in everyone. So it seems likely that the gut microbiome might be involved. If you just take normal NASH patients, not those that have had obesity surgery, and measure through breath tests whether they've got small intestinal bacterial overgrowth, more of the ones with NASH have evidence of small intestinal bacterial overgrowth than those who've just got steatosis. As again, I'm sure you know, in an institute like this, the main culprit in gut microbiome of trouble is endotoxin. So endotoxin in the wall of gram-negative bacteria gets absorbed into the portal vein, gets up to the liver, and starts to activate receptors, including TLR4, on Kupffer cells, which causes them to release inflammatory cytokines. And if you produce a mouse with a knockout of the TLR4, the endotoxin receptor, you can make it as obese as you like and feed it with oxidant-deficient diets, etc., and it will never get NASH. So it would seem that if you take out the endotoxin pathway, you protect a mouse anyway against NASH. We know that one of the reasons you get endotoxemia in the portal veins in alcohol patients, we've known for years, is because alcohol increases gut permeability to endotoxin. And we've now got some evidence that you see exactly the same thing in obesity. So it would appear that you've got evidence of bacterial overgrowth. You've got evidence of increased permeability, which will allow the endotoxin through. We and others have measured endotoxin, as they have done in alcohol patients, and found that in NASH patients, you get high levels of endotoxin in their peripheral blood compared to those patients without NASH. There's been some recent interesting data on taking patients with NASH, measuring their microbiome with genetic techniques, subtyping the microbiome, and being able to group patients' NAFLD severity with their microbiome subtypes. And this is the first real good human data starting to show that this thing here might be influencing fatty liver. And then there's this fascinating study that was produced in Nature early last year, which suggests that you can catch NASH by acquiring someone else's gut microbiome. And the data from this are here, the relevant data. So this was producing some mouse models of NASH by knocking out a couple of cytokine regulators in the inflammasome and creating a nice mouse model of NASH. It also, though, affected their ability to fight gut bacteria. So these two mice with ASC and IL-18 knockout developed bacterial overgrowth and NASH. So that's if you just take this one here, this is the NASH that they both developed. But what was interesting was that when you bred a wild-type mouse in the same cage as these bacterial overgrowth mice, the wild-type mouse got raised AST and got NASH as well, so and acquired the gut bacteria. So it seems to be that if you get these nasty gut bacteria from another mouse and you acquire those, then you can essentially acquire NASH as well. So it would suggest that NASH, possibly in humans, might be a transmissible disease. So a growing body of evidence that our microbiome, either the one that we're born with or the one that we develop with time, might be determining who gets NASH and who doesn't. And of course, again, that opens itself to treatment. So it's a very attractive idea that it may be something to do with our gut microbiome. So now I'll move on to what's obviously more my own personal research interest, genetic factors. And what is the evidence that there may well be a genetic factor controlling this? Well, I'll show you some data in a minute. It's very clear that NASH and NASH fibrosis and cirrhosis cluster in families. Now that, of course, could be because those families have shared environment risk factors, they all eat too much, or they have shared genetics for diabetes and obesity. But it certainly aggregates in families. If you look at twin studies, which I'll show you, there's a good correlation in monozygotic twins of serum ALT, suggesting that we inherit genes that determine our liver's response to insult. And then virtually every study that's looked at different ethnicities have shown that, particularly in North American studies, that Hispanics seem to be more at risk of developing NASH than white Americans, who are more at risk than black Americans. And that doesn't seem to be explained solely by the increased risk of diabetes in the Hispanics. So you've got three bits of evidence there that NASH might have a genetic basis. And here's the familial aggregation study of Schwimmer and colleagues looking at your risk of getting NAFLD if one of your siblings had NASH, 60% as opposed to 10% if they didn't. And if one of your parents had fatty liver, you were 80% likely to get fatty liver as opposed to only 35% if you didn't. And when they looked at correlations between body mass index and liver fat fraction, they found a lovely correlation in NAFLD children, but not in no NAFLD children, again suggesting there's something familial going on here. They calculated that the heritability of fatty liver was 100%, and of the fat fraction, the amount of fat in the liver was around 40%. This is the twin study looking at twin A versus twin B in MRI studies done in Finland in Jikki Järvinen's group, showing again if you're monozygotic identical twins, a very nice correlation between twin A and twin B of 0.65, whereas if you're only if your dizygotic twins only share half your genetic makeup, the correlation is much weaker, suggesting again that there's something you inherit that determines your liver's response to an insult. So we've got some reasonable evidence that susceptibility to fatty liver is genetic. Why bother? I'm often asked about this. So you find the genetics, but so what? Because the patient's got those genes, you can't alter them. So why try to understand the genetic basis? Well, I think there are at least three reasons. So the first one, obviously, if you can identify genes that predict risk, then the products of that gene obviously must be involved in pathogenesis. Somewhere it might help you understand the pathogenesis and help you develop new treatments. It may well be, just like in the 60s, when I didn't start in the 60s, thankfully, but in the 60s, hepatitis was one diagnosis, and everybody got some steroids. We didn't know that hepatitis was hepatitis B, in some patients hepatitis C, in others NASH, in others alcohol, in others. And so the idea that I suspect that NASH, the liver only has very minimal ways of responding. It can either get chronic hepatitis, like in these immune conditions, or it can get necrosis, like if you take too much acetaminophen, or it can get fatty hepatitis. And I suspect that we know alcohol versus non-alcohol as two causes of steatohepatitis, and some drug causes, some drugs like tamoxifen that cause it. But I think even in those fatty liver patients, what we'll ultimately find is that some patients it's driven by a particular pathway, and in others it's driven by a different pathway. So so-called stratified medicine, precision medicine, etc., will be diagnosing patients with NASH and then using their genetics to say which pathways are important in them, and that becomes then relevant to which treatments you give them. And of course, the obvious one of why I understand the genetics, if you could identify that you are particularly at risk of developing advanced liver disease if you drink or if you get overweight, you would counsel them early on and give them, hopefully, good prevention strategies, because you know they're the ones at risk. So how do you go about finding the genes? And I guess a lot of this will be familiar to you, but you know, you do what we've done for years, is you think, well, I think TNF-alpha is important in NASH, so I will look at genetic variations, SNPs in TNF-alpha in cases with advanced NASH and compare them with cases with steatohepatitis and see if they're different. And that's a classical candidate gene case control study, and that's what we relied on for the first 20, 30 years of the field, really. I guess we started to see the first candidate gene studies coming out in liver disease in the late 90s, probably early 90s. So I guess around 20 years of this has been largely candidate gene case control studies, and I'll talk to you about those. And then what we're seeing more and more now, of course, is a hypothesis-free or generating approach where we look at it across the entire genome at the moment, largely with SNP chips that look at thousands of SNPs across the whole genome, but increasingly what we'll be seeing in the literature is whole sequences of patients as the price of doing a whole genome sequence comes down. It's now at about $1,000 per patient. You can get a whole genome sequence now. Okay, so what about these candidate gene case control studies? Have they told us anything? Have they produced anything useful that we can rely on? Or are they all just false positive type 1 errors? If you do enough SNPs in your cases and controls, you'll find something that's different and you'll get your paper into a journal. But it's not really an association. Is that what we're left with? Well, I think journals like Nature Genetics and others got very quickly onto this, that actually if you did enough SNPs in enough patients you would find some differences. So the community started to come out with rules really that said this association between a SNP and disease is probably real, whereas this one could well be spurious or by chance. And so I think the community started to develop a way of saying this is probably all right. And I think if you fulfill the following criteria, it's probably a reasonable, strong association that you can rely on. So the first one is that the SNP that you're looking at, you've got some evidence that it actually does something to either gene expression or protein function. It affects the catalytic site of an enzyme, or it sits in a gene promoter and affects gene expression. So have you got evidence that that polymorphism does something to gene expression or protein? Have you been able to replicate that association in a completely separate group? Because obviously that again gives you confidence that it's not just a chance association. If you get it by chance twice, that starts to reduce the likelihood that this is spurious. TD testing is something that we've relied on very heavily in Newcastle. And what that says is, you know, if this particular SNP, this particular allele is really the risk allele, what you want is heterozygote parents. In an ideal study, you don't need to know whether the parents have disease, but you would like one parent to be homozygote wild type, and you'd like the other parent to be heterozygote for the risk allele, presumably less common allele, and the normal allele. And then you look at the affected children. So you've got your young adult with NASH, and you look to see how many times those with a heterozygote parent, the risk allele has been transmitted to them. So if this is just random, you would expect 50-50, wouldn't you? You'd expect half of the affected kids to have got that affected allele and half of them not to have got it by chance. The further away it gets from 50-50, the more, which is called preferential intrafamilial transmission, again, you can test the significant difference from 50-50 with a classical chi-squared test, and it gives you confidence that this is likely to be a real association, and I'll show you some examples of that in a moment. And then, so if we look for association studies in NASH that are out in the literature that fulfill these criteria, they've got evidence of function, they're replicated, or they've used familial testing, what are we left with? And we're left with only 4 genes. And I'll give you the data on this in a moment. So KLF6, which is a fibrosis gene, it controls collagen gene transcription in stellate cells. 2 insulin resistance genes, makes sense because of the role of high levels of insulin in driving fat and inflammation. Superoxide dismutase 2, an enzyme very dear to Jose's heart, which is the mitochondrial antioxidant enzyme. Something called SLC2A1, which I'll talk to you about in a minute, which has come out of some work that Jose's been involved in. And I just put in a little mention of ApoC3, and you'll see why in a moment. I could have put lots of knots here, obviously, for all the many, many studies that are out there that were never replicated, that had no functional basis. But I have to mention ApoC3 because I'm usually asked about it for a particular reason. So here's the KLF6 data. So KLF6 was in gastro a couple of years ago. So KLF6 is this stellate cell transcription factor. There's a polymorphism in it, which creates an alternative splice site, a G2A, and you'll see in 3 hepatocyte cell lines, when you've got the G2A, you get these multiple splice forms. And these act as negative dominant mutants. They bind the transcription site, and they stop the normal KLF6 binding normally and activating transcription of genes including collagen. And here's some functional data down at the bottom here, where you see that when you express in these hepatic stellate cell lines the G2A KLF6 gene, that you get much less collagen 1 and alpha smoothosylactin than you do in the wild type. So you've got evidence of function. This was the data in our UK cohort, in a separate Italian cohort, and then in both put together. And what you find is, if you have this polymorphism, you would expect it to be protective, because it is not allowing KLF6 to activate the transcription of collagen and other fibrosis genes. And sure enough, if you have mild fibrosis in our studies, you're much more likely to have this polymorphism than if you have the wild type. So the wild type is the dangerous one, because it's the one that allows KLF6 to function properly. And when we looked at this in multivariate analysis, we found the usual things are associated with increased risk. So age, how heavy you are, whether you're diabetic, whether you've got a low platelet count, low ALT ratio. But the KLF wild type gave you a risk odds ratio of around 3 of having advanced disease than if you didn't. And then to finish off, we managed to find, working with Valerio Nobili in Rome, who has lots of children collected with fibrotic NASH, we were able to get what we call trio DNA, where you've got the DNA from the affected child and both of the parents. And when we found these heterozygote parents, 24 of them, we found that 17 had transmitted the wild type and 7 had non-transmitted it, which is significantly different from 12-12, which is what you'd accepted. So it was reproduced, it was functional, and it preferentially was transmitted to affected children. Here's the insulin-resistant gene data. So here's the functional data first. So this is looking at phosphorylation sites of AKT, which, as you know, is downstream from insulin signaling, and showing that whether you look at the phosphotyrosine 308 AKT in white here, that if you have these loss of function polymorphisms in IRS1 or ENPP1, which are both involved in the insulin signaling pathway, or phosphorylation of AKT, at that different site in black, you get this decreased insulin signaling. So we've got evidence that these genes, as you might expect, have been associated with diabetes. These polymorphisms, they're functional. And when you look, again, at UK cohort, Italian cohort, what you find is that the percentage with advanced fibrosis, so more than stage 1, most of our data looks at stage 1 and 0 together because it's the odd little wisp of fibrosis, but significant fibrosis we count as 2, 3, and 4 because it means you've got bridging, you've got cirrhosis, etc. If you have wild type, you've got around a 40% risk in our cohort. If you have one of the SNPs, it goes up to 50. If you have two, it's up to 70, and you'll see you get exactly the same pattern in the Italians, and obviously when you put them together. So again, replicated and functional. This is the data on SOD2. This is the famous alanine-valine SNP. It's the key mitochondrial antioxidant. The C to T polymorphism encodes an alanine-valine variation in the mitochondrial targeting sequence. So importantly, it affects whether this enzyme, which is nuclear transcribed, then gets into the mitochondria where it does its work as an antioxidant. The alanine, or the C allele, is better at getting into the mitochondria, so suggesting that the T allele will be the risk one because it doesn't get into the mitochondria. When we did the TDT test, this time, because it's a more common polymorphism, we had 55 informative families that had a heterozygote parent, and the T allele was transmitted almost twice as often as not transmitted, which is significantly different from 50-50. And when we looked at again, the percentage with stage 1 fibrosis, when we looked at the wild-type homozygote, the heterozygote, and the homozygote rare T allele, you'll see that the risk goes up in a straight line in both UK and Italian cohorts. And finally, this is the data that Jose has been involved with, which I came across last week, which I think is very convincing. So this is solute carrier family 2 member 1. 2 SNPs were associated with NAFLD in 2 independent cohorts. So you've got your evidence that it's not just in one cohort. One particular SNP, they showed the functional effect, that it was associated with reduced mRNA expression in these liver biopsies. They also showed that mRNA expression went down generally in NAFLD patients compared to controls. And when they silenced the SLC2A1 in vitro, they found that when exposed to stress, such as oleic acid, these cells develop more fat and more oxidative stress. So I think this can be added to the list of genes that look to have reasonable evidence that they may well be important predisposing factors. This is why I have to mention ApoC3, because of this striking paper that appeared in the New England Journal of Medicine a couple of years ago. It had an editorial in it, and it said, this is the cause of NASH. That if you are wild type, this was liver triglyceride content in Asian Indian men measured by MRI. If you are wild type for the ApoC3, remember ApoC3 is a key apolipoprotein that's involved in VLDL synthesis. If you don't have ApoC3, you can't make VLDL and you can't export fat out of the liver. So it would make sense that if you had a defective ApoC3, that it might be bad. And so you can see the amount of liver fat in these variant allele carriers. What this demonstrates, however, is the small number of dots on this diagram. And this is classical type 1 random finding error. It's got a very, very famous diabetes doctor at the end of it called Jerry Shulman, who regularly publishes in Nature, Cell, and New England Journal. And I can only assume that having his name on the paper is how they got this very simple candidate gene study into the New England Journal. Because here we have 1, 2, 3, 4, 5 studies I'm aware of at least. So this was ours, 758 Europeans. Finnish study, 417 Finns. American study, 2,500 Americans, and another American study with 455 children. Not a suggestion of any difference in ApoC3 polymorphisms in those with fatty liver and those without. And so I think, you know, when you get asked about it, everyone knows about this paper because it was in the New England Journal, but it just hasn't been replicated, and I think it's essentially spurious. So the final part of my talk really is where we are today. What have we been doing around GWAS, where we don't have ourselves restricted by our knowledge of what might be involved in pathogenesis and looking at a polymorphism in a particular gene. We look at all the genes, and this of course is what's changing the field of genetics, and there is now a bunch of emerging data in NAFLD that are pointing us in new directions. So I guess for those of you who aren't geneticists, the principle of genome-wide association studies is fairly straightforward. You get a whole number of unrelated cases and match controls, and you look to see if any polymorphisms in any of the genes that you're looking at are more present in the cases than the controls, and you do a classical p-value odds ratio. The problem is that compared to a single candidate gene study, your p-values have to be much, much smaller. Because obviously the chances of a random association when you're looking at 500, 600,000 SNPs at the same time are very, very high. So you've got to have p-values of 10 to the 20, 10 to the minus 20-ish before you can be confident. So where have we got to with NAFLD? So there have been, before the data I'm going to tell you from the collaboration I've been involved in, there have been 4 attempts at GWASs in NAFLD. So the first one, Nature Genetics paper, real paper, paradigm-shifting paper this. The diagnosis of fatty liver was made just with MRS, so there was no biopsy, so it couldn't tell you anything about my question, which is who gets advanced disease versus mild. It could just tell you who gets fatty liver and who gets worse fatty liver, more steatosis than others. So 2,000 plus patients, they looked at 9,000 SNPs, and they found, and I'll show you the paper in a moment, this gene PNPLA3, which encodes adiponeutrin. And that's been the game changer, really. It's a perfect example of what you get out of a GWAS. Nobody would have ever done a candidate gene study on PNPLA3, but it came out of this GWAS. There was then the first histological attempt. This was from the NIH CRN network, where they did a GWAS, but in only 236 patients. They did have histology, and a whole variety of what it looks like are spurious associations came out, because if you've only got 230 patients and you look at 324,000 SNPs, you're bound to find differences in that small number. There's no replication, and I think I'm not quite sure what that means. The big worry about this data is it never even showed up PNPLA3, which is about the strongest risk factor there's ever been in any liver disease, in fact in any common disease. So I think we can just sort of put a little dotted line through that, but give it some credit for being the first GWAS done with histologically defined NASH. You've then got a funny paper which was really CT steatosis taken from many, many different studies that had looked at things like cardiovascular mortality, lipids, lipid levels, and these previous studies had CTs of their liver and steatosis classified, and so they had a lot of patients and they were able to essentially reproduce the Romeo study. They found a strong association with PNPLA3, but also some other associations. And then until the data I'm going to show you, the biggest histological study published just late last year, a Japanese study, nearly 500,000 SNPs, and again, PNPLA3 came out. So it looks as if we've got definite PNPLA3, and we might have some other genes which need replicating. So this is the game-changing paper of Romeo and colleagues in Nature Genetics. So they had over 2,000 patients, nicely split into Black Americans, White Americans, and Hispanics. So it also addresses the question of why is it that Hispanics get fatty liver more than whites, more than Black Americans. So they did a genome-wide association, non-synonymous SNPs, so they only picked SNPs that changed amino acids. So that's why it was less than the usual 500,000, because they were very targeted. But making the point again that this was MR, magnetic resonance defined fatty liver, so they couldn't say anything about steatohepatitis. This is the result. So they have one SNP that comes out. As you know, this represents all of the 23 chromosomes, and this is the log to the -10 of the p-value. And what you're getting is a p-value here of 10 to the -10 for this PNPLA3 gene, with having the G allele associated with an increased hepatic fat. We don't really know, and I'll come on to this in a moment, exactly what PNPLA3 does, but it seems to have at least some phospholipase activity. So the assumption has been that it might be a loss of function phospholipase with your G allele that means you can't break down fat. This was the data from the paper, more data, which shows very nicely why it is that Hispanics get more fatty liver than blacks and whites. So in each of these three groups, if you're CC, you have less fat than CG and less fat if you have GG. But here, the Hispanics have a much higher frequency of the GG than the others. So it also explains the ethnic difference. What it also shows is that the mechanism of this effect is not through insulin resistance, it's not through an effect on obesity, and it's not through lipids. So there was no impact of the PNPLA3 polymorphism on the classical risk factors. It didn't make you fatter, it didn't make you more insulin resistant, and it didn't raise your, affect your lipids. So here's our data. So this has been presented at the AASLD in November, and it's about to be presented with further analysis at EASL in the plenary session in Amsterdam in a few weeks' time. So what we had was a large consortium funded by the EU called FLIP, which was for fibrosis, liver injury progression. Fatty liver indicators of progression, sorry, I forget what FLIP stands for. You'll see we've got 3 centres in the UK and centres from all over Europe who basically have pooled their patients with fatty liver disease, samples, liver biopsies, DNA, sera, etc., to address a whole range of different questions. So we defined you had to be European Caucasian, you had to have alcohol limits as we talked about, you had to have a biopsy that had been scored by the Kinase system, which gives you a NAS score, which is the NASH activity score, which is made up of steatosis severity from 0 to 3, ballooning severity from 0 to 3, and lobular inflammation. So you can get a score of between 0 and 8. And to do our multivariate analysis, we were only interested in patients that had BMI, waist circumference. We knew whether they had diabetes and metabolic syndrome. We had some standard liver blood tests. We knew they didn't have other liver diseases. And they hadn't been taking drugs that might have affected the amount of fatty liver they had. So we had over 2,000 patients, all with histology. We divided them into a FLIP GWAS cohort of over 1,000 patients, and then a validation cohort, so that anything that came up in the GWAS, we could then go back and check. We also had data that from that paper I showed you of the 236 patients with a GWAS published in Gastro from the NASH CRN. And we used a similar chip, so we were able to pool their data with our data to get 1,125 samples with histology and anthropometry and BMI and everything else. We compared the frequency within groups, obviously, fat versus no fat, fibrosis versus no fibrosis, but we also compared the frequency with a standard Wellcome Trust case control consortium population controls of 6,000. We used the Illumina OmniExpress chip, 733,000 SNPs, and it captured 91% of those SNPs that had a frequency of more than 5%. Here's the data, so it looks like a sort of familiar type of demographic, age around 50, mostly women, BMI around 37, about a third of them diabetic, nice spread of steatosis severity, ballooning severity, fibrosis severity. So we were able to ask the questions, did any of these SNPs associate with advanced disease? And this is the results that we got. So this is comparing severe steatosis, so grade 2 or 3, versus controls and those with hardly any steatosis. What you will see here essentially is PNPLA3 coming out largely. That's the big effect. But also a whole variety of other SNPs that had smaller effects, p less than 10.6 10 to the minus 5 here. But the big effect here was around PNPLA3 again. When we looked at NASH versus controls, we got PNPLA3 again, but also these other genes here, which seem to be having significant effects. And when we looked at fibrosis again, we get PNPLA3, but these other genes around here with smaller effects. When we then went on to validate this by looking at those SNPs and seeing if any of them came up again in the 800 validation set, essentially, I hope you can see this slide, we get, well, you can because I've put them in blue, you will see that there are essentially 3 genes, PNPLA3, glucokinase regulator, and TRIB1, which associate in the validation set with both fat, NASH, and fibrosis. So all 3 parameters are associated. Then there's another few genes that are just associated with one of the parameters. Most of the things that came up in the GWAS weren't replicated. So glucokinase regulatory protein regulates glucokinase, which is involved in hepatic glucose metabolism. It's the enzyme that phosphorylates glucose and makes it into glucose 6-phosphate in the liver. It's the rate-limiting step of taking glucose up. And the GCKR polymorphism seems to be loss of function. So you get more glucokinase activity, more precursors for fatty acid synthesis through the glycolysis pathway, but also a depletion of ATP because you're madly phosphorylating glucose. And that, of course, could be deleterious and lead to injury. TRIB1 encodes Tribble's homolog 1. It regulates hepatic lipogenesis and VLDL production. Previously, SNPs in TRIB1 have been associated with myocardial infarction and serum lipid levels, so it makes sense that something around TRIB1 might be also causing NASH and fatty liver as well as PNPLA3. There have been a number of candidate gene studies after the PNPLA3 study came out that have replicated this was the first replication study, 291 FINs, showing with MRI that the more G allele you have, the more your liver fat goes up and the more your serum AST goes up. This was one of the studies we did with the Italian group looking to see whether PNPLA3, it was the first study that looked to see whether PNPLA3 predicted risk of advanced disease, not just whether you get NAFLD. And here again you will see whether it's presence of NASH or presence of fibrosis. The GG patients in Italy, UK and combined had more NASH and fibrosis. So you definitely, as confirmed in the GWAS, PNPLA3 and GCKR and TRIB1 don't just make you more likely to get fatty liver if you're overweight. They make it more likely that you get inflammation and fibrosis. When we looked for the TDT test, 71 informative families, child with fibrotic NAFLD, the G allele was transmitted twice as often as not transmitted, which was much more significant than, highly significantly different from 50-50. And there have been probably 20 studies now looking at PNPLA3 in candidate gene studies. Most recently, it would appear in alcohol, and we'll talk a bit more about this tonight, that PNPLA3 is also associated with the risk of alcoholic liver disease and indeed of hepatitis C liver disease. And if you look at these now, 1, 2, 3, 4, 5, 6 studies that have looked at the role of PNPLA3, this is the polymorphism, and risk of liver cancer in ALD, you will see that the PNPLA3 is associated with, in some studies, just with ALD and cirrhosis, which is what you would predict from the NASH studies. But a couple of studies at the bottom here showing it's also associated with the risk of developing liver cancer. And so we've recently looked at this, and again, this is being presented at EASL in a couple of weeks' time. We found that the frequency of the GG homozygote, this is the rare allele here, was much more common in NAFLD with liver cancer patients than in normal NAFLD patients. Again, suggesting that this PNPLA3 is doing something to predispose to inflammation, to fibrosis, and also to liver cancer. Here's some more recent data we've been looking at in collaboration with Naga Chalasani's group, which is expression QTL analysis. So this is saying, okay, well, the SNPs that we've got, what about seeing if any of the SNPs that are on our chips actually correlate with expression at liver mRNA level with PNPLA3, or anything else you might be interested in. And this is what expression quantitative trait loci analysis is called. So what they did here was take a whole bunch of livers. They measured the mRNA expression in those livers, and then on those same patients, they did a GWAS. And they pulled out all of the various SNPs in PNPLA3 and said, did any of those correlate with expression of PNPLA3? And what you see here is that there is a SNP here which has 10 to the minus 6 effect on expression of PNPLA3 clustered around a group here. There seems to be other SNPs that are also associated strongly with expression. This is the 148M1, which you wouldn't expect to be. So there's no evidence of this affecting expression, because remember that polymorphism affects the amino acid sequence around the active site. So that's not doing it via expression. And then because of our collaboration, we were able to say, well, do any of these additional SNPs in PNPLA3 that correlate with PNPLA expression, do they associate with NAFLD? And in our FLIP cohort, this one here, the one that's most strongly associated with expression of PNPLA3, is also strongly associated with fatty liver, steatosis, steatohepatitis, and fibrosis. We've identified additional regulatory SNPs in PNPLA3, as well as the functional amino acid changing SNP that's also associated with NAFLD, giving you more evidence that PNPLA3 is so critical in determining who gets this disease. So why? Why is PNPLA3 associated with fat, NASH, and fibrosis and liver cancer? And the short answer is we don't know. I'll show you some functional data that's come out which suggests it might be doing both of these things. So there's some evidence that PNPLA3 is involved in triglyceride hydrolysis, and that the loss of function in this key SNP leads to less hydrolysis of triglycerides, so you get more fat in your liver. There's also some evidence that it might increase an activity of an acyl transferase and increase lipid synthesis or fatty acid esterification. So there's two reasons based on PNPLA3 function why a SNP might give you more fat. But of course, neither of these things explain, just because you've got more fat in your liver, why you would get more inflammation and fibrosis. So here's the data from JCI published in around August last year, where they overexpressed the PNPLA3 polymorphism in a mouse, but only in the hepatocytes, because they linked the PNPLA3 transgene to an albumin promoter. So when you express PNPLA3 polymorphism in hepatocytes and then feed them high fat, the ones with the polymorphism get more fat in their liver, here shown histologically, and here shown quantitatively cholesterol esters, triacylglycerol in red here. So there's no doubt you get more fat in your liver if your PNPLA3 is the G allele. But when they looked at levels of TNF-alpha message, collagen message, smooth muscle actin message, there was no difference. So if you overexpress that PNPLA3 polymorphism in hepatocytes, it doesn't give you more NASH and fibrosis. So what does that mean? Does it mean that in these mice, to get the progressive damage, you need a second hit? You need a bit of endotoxin, you need some source of oxidative stress, and that you wouldn't expect these mice to get anything other than fatty liver if all you do is give them a bit of high-fat diet? Or is PNPLA3 affecting other cells? So is it involved in stellate cell function or inflammatory cell function? If you just overexpress the PNPLA3 in hepatocytes, you won't see these effects. And I think perhaps the most fertile area for study in this area at the moment is to work out the mechanistic links between the metabolic genes, PNPLA3, GCKR, and TRIB1, and fat, and progressive disease. And if you want a simple answer, I think what's happening is that these pathways, such as the glucose-6-phosphate pathway, they're very high up in the metabolic scheme, and perturbing them will have different effects. One might be giving you more precursor for fat in your liver, which is separate, and then another one might be affecting, for example, ATP levels or inflammation levels. And I think they're doing two separate things. My last couple of slides are about epigenetic factors. So I'm sure you're aware that epigenetic DNA modification, e.g. by methylation or acetylation, so this is not your genetic code, this is methylation of your genes that in the transcription sites that stop those genes being transcribed. It probably explains why maternal obesity is associated with adverse fetal outcomes. So whether your mother, in mouse models anyway, and probably in humans, is fat, determines your risk of developing fatness or diabetes. In humans, if you breastfeed, that seems to protect you against the development of NASH. So something about being fed with breast milk early in life determines your risk of getting NASH. In mice, if you high-fat feed the mother, that affects your adult offspring risk of developing NASH. And in mice, fibrosis risk is transmissible through generations. So if you give mice a small dose of carbon tetrachloride so that they get a bit of fibrosis in their liver, and you then breed those mice and give the offspring carbon tetrachloride, they get less fibrosis in their liver, and their offspring get less, and by the time you get to the 4th generation, the same dose of carbon tetrachloride that caused aggressive fibrosis in the great-great-grandparents causes nothing in the 4th generation. And this is a paper from our group in Nature Medicine last year, which showed that. What it showed was that you transmit methylation through sperm of fibrosis genes in the liver, so that your offspring remember that your liver was exposed to fibrotic stimuli and switch off the fibrosis genes. And what we did, because the editors asked us to, was to get some human data, was we looked at methylation of PPAR gamma, which is an anti-fibrotic gene in the liver, to see whether that had been switched off more in those with advanced fibrosis in NASH. And here you get those with advanced fibrosis had more methylation of their PPAR gamma gene, so switching off this antifibrotic gene, and in fact they had less methylation of TGF-beta, the profibrotic gene. So to conclude, the risk of progressive NAFLD is probably due to environmental and genetic and epigenetic factors. Diet, exercise, and coffee, probably obstructive sleep apnea and gut microbiome are the most likely environmental factors, but they've all been studied relatively little. Several PNPLA3 polymorphisms, it looks like, not just the classical one, are associated with fat and progressive disease. And the question now is this ready for the clinic? Should we be using PNPLA3 to determine who to treat with what? Are those patients that have got the polymorphism less likely to respond to diet and exercise, for example? And we're just starting to see some early studies around that. There are some other associations come out of at least 2 GWASs now, and understanding the mechanisms of those will become important. And finally, it looks as if parental factors, through these epigenetic factors, may well be determining who gets disease through modification of gene expression. Thank you.
Any question or comment? Chris, is there any relationship between PNPLA3 and the risk to develop injury in response to drugs, I mean, DILI or any other gene? Yeah.
So in terms of DILI, there has now been GWAS studies done in DILI, as you know, and the only things that have come up with different drugs are HLA, essentially. So we've done studies with flucloxacillin and augmentin. They're different HLA types, but they're all HLA associations, suggesting that both those illnesses, at least those, are immune conditions. So certainly I'm not sure of any study that looks specifically at PNPLA3, but it will have been in the GWAS SNPs. So at the moment, it's getting numbers, of course, as you know. I mean, in terms of drugs that cause fatty liver, you'd be looking at tamoxifen, something like that, and to get enough cases that would enable you to do the study. So in terms of GWAS of DILI, it's just been HLA types that have come out so far. Same with the autoimmune liver diseases, it's been HLA, as you would expect. Yeah.
Nice talk. About the epigenetics changes in the NAFLD disease, have you measured or do you have any idea about the SAMI levels in that livers?
No, we don't, but I'm sure we could do some collaboration and measure it. Yeah, no, I'm not aware of anybody that's done that yet. Are you? You'll say that's measured actual SAMI levels? No. As you know, we're very interested in giving SAMI as a treatment for NASH, given its antioxidant and other positive factors. But I mean, there are polymorphisms in the MAT genes, aren't there? And you know, and they don't come out of any of these GWASs yet. Has there been much candidate gene work done on MAT?
Well, we can talk to the... Yeah. Anna was doing those studies. She can tell you later, but I think she didn't get much. One question, you mentioned in the introduction about fructose. Can you say a word about fructose?
Yeah,
I think that people will be interested about fructose.
I mean, so it makes biological sense because, as you know, fructose doesn't require phosphorylation. It just goes straight into the liver. So there's no rate-limiting step. Whereas with glucose, if you can't phosphorylate it, essentially dams up and blocks transport in. So the more fructose just bypasses any regulation. The more fructose you eat, your liver just takes it up and converts it into fatty acid precursors. So it makes sense that fructose would be bad for fatty liver. The problem I understand from these studies is they've all looked really, and they're all American, and they've looked at the amount of corn syrup that is in the diet. And the problem with corn syrup, as I understand it, is that it's 50-50 fructose and glucose. So what that probably means is, so it's been headlined as fructose is dangerous, but it probably is just calories intake. So I examined a PhD thesis recently from a nutrition group, and they took these papers apart and said, actually, there's no evidence that this is fructose, it's just sugar. Because it's very difficult to take fructose without, the same amount of glucose. So I think the theory is good. I'm not sure why it causes fibrosis, which seems to be the association. And I think the association is largely because the more fructose you eat, the more stuff you eat, the more calories you eat, you're more likely to be obese and insulin resistant. I don't think as yet we've got data that says it's definitely fructose that's important. Of course, it's not used in Europe hardly at all. We don't use corn syrup here because we don't have to keep the farmers in North America, in profit, so we don't use it. So it's not as big an issue here. If they took corn syrup out of the diet in America, the whole farming industry would collapse. But we don't have it.
I have a final comment then. Bariatric surgery is now common.
Yes.
But morbidly obese people, most of those could have fatty liver.
Yes.
Is there anything known about the effect of bariatric surgery and this drastic weight loss into
Yes. So
fatty liver evolution?
Yes. So it's without doubt the most successful treatment. So there are now for obesity, as you know, and there are now 5-year follow-up studies done by Philip Maturin predominantly. So the early studies with short-term follow-up all showed that 95% of patients lose the fat in their liver, and it seemed to the amount of fat you lost seemed to correlate with the degree of improvement in insulin resistance. So you lose all that weight, you become insulin sensitive, and your liver clears of fat. The longer studies have gone on, you're now starting to see resolution of inflammation and even fibrosis. So it seems that if you take away the free fatty acid drive, the insulin resistance, presumably therefore the oxidative stress that's been as a result of the fatty acids being metabolized by these alternative pathways, it seems to be the best treatment. And of course, what we're trying to do is we can't have obesity surgery for it's 40% of the population in the UK, I don't know what it is in Spain. So we're going to have to come up with some medicines really that will do that effect. But certainly it works. If you lose that much weight, your NASH and fibrosis vanishes. It's very good.
Don't know any other question, comment? Thank you very much.
All right.